there are a few agents right now that are kind of making their way through some very, very early stage. So can you guys just discuss, any of the major agents or clinical therapies that are currently in the pipeline right now that seem promising to promote remyelination and, what sort of sets them apart mechanistically or clinically from each other?
Yeah, I mean, I can start there. I think there's been many academic and industry studies that have identified therapeutic approaches to enhance the regeneration of oligodendrocytes and promote remyelination. I think much of this work has emanated from drug screening efforts on oligodendrocyte progenitor cells, which we talked about earlier, have this remarkable capacity to regenerate new oligodendrocytes in the central nervous system. Right. And collectively, these studies have revealed and validated, I think nicely a handful of druggable targets that both drive oligodendrocyte regeneration as well as remyelination. And so in my own lab around with along with my Case Western Reserve University colleague and Convello, a therapeutics company that I co-founded with Drew Adams, we focused our own efforts on two enzymatic targets in the cholesterol biosynthesis pathway, namely EBP and CYP51, which, when inhibited, these drive the accumulation of a bioactive sterol that promotes the generation and survival of new oligodendrocytes and remyelination. And so if you analyze all the screening data that's been published by many labs and company, it's clear that many molecules targeting EBP have been identified as a very prevalent class of hits, maybe the most prevalent class of hits. Now, there's other exciting potential targets that have been identified and are being pursued. Ari will certainly talk about the muscarinic M1 receptor, others including GPR17, thyroid hormone receptor, some of the receptor tyrosine kinases. These have been identified and are being pursued by a number of academic and industry efforts. But I think what's important to remember about this is that the regeneration of oligodendrocytes and remyelination, this is a multi-step developmental process, right? This is not simply one cell divides into two and now you've replaced a cell. So the remyelinating drugs that have been identified and are in development across multiple programs, I think are each likely targeting slightly distinct stages or aspects of this regeneration or disease process, right? Some are stimulating directly OPC differentiation. Others are promoting the maturation of immature oligodendrocytes. And some are enhancing the survival of newly formed oligodendrocytes, which I think is a key bottleneck in vivo to ultimately generating enhanced remyelination. And I think lastly, others are, overcoming sort of local disease-specific barriers in the lesions and in other places that are preventing regeneration, so-called approaches to remove the brakes. And I think I'll just sort of say in closing here that this mechanistic diversity among the current therapies is really important, right? We don't know which one's going to work, but ultimately, I think each of them will work on different stages of the regeneration process, and they may sort of ultimately complement each other, rather than compete. And it's possible that different stages of the disease, or there might be different patient responsiveness to each of these distinct remyelination therapies. And I think, based on the underlying disease stage or biology or genetic factors, might prove more amenable to remyelination by each of these agents.
Yeah, I would only echo that I think there's a lot of different stuff that is exciting. As Paul touched on, we've been excited by targeting the M1 receptor and antagonizing the M1 receptor as a means of inducing OPC differentiation of oligodendrocytes. I think there are some of the approaches that have been taken that are way more robustly and demonstrated and replicated in multiple laboratories. So I'd encourage people to look at, which approaches are replicatable and have shown, capacity in a lot of different contexts and settings for remyelination to occur. I think I'm hopeful that a few different approaches will be successful in working. I do think people have to be cautious. There are claims that are made about approaches, some of them that are nutritional, some of them that are using elemental approaches. that are claimed to have a quote unquote remyelinating effect with very little biological evidence that they in fact do. And I think it's super important for the field. None of us likes to denigrate anybody's work, but we have to be, in the interest of patients, we have to be very clear about the limited amount of data that supports claims for remyelination for some of these approaches. And people have to be cautious and wise about interpreting that. We also have to be cautious. I mean, our own experience would teach us. We chose to pursue the M1 approach. We partnered with some folks in industry. The challenge there is that the folks in industry have a different set of priorities from those of us who are doing scientific research and who are involved in clinical care. I mean, their obligation is to people who are investors, and not necessarily to the development of drugs that are successful therapeutically in the ultimate aim of helping patients with MS. So, I think we have to be both wise on the side of judging the science, and we also have to be wise on the side of and learn about how we can advance the ball without necessarily handing over the keys to people whose interests don't align with those of us who really care about MS.